So we are interested to particularly focus on studying the clinical behavior of the ER-positive and HER2-positive breast cancers, as well as learning the biological features of these triple positive breast cancers by performing comprehensive molecular characterization of these samples. So what we have done is performing comprehensive bulk-based multi-omics assays on some very extraordinary interesting cases, as well as by integrating this data with the emerging techniques, spatial biology data to study the intratumor and intertumor heterogeneity. So what we find that among the HER2-enriched subtype, TP3 mutation naturally is associated with good response. But if we remove this interesting HER2-enriched good response cases, TP3 mutation is actually associated with poor response, which is considered ER-positive HER2-negative tumor. Now, when we start to integrate this data with the spatial whole transcriptomic and proteomics data, we start to notice they are very different in terms of each of the tumor regions when we map with the tissue samples. Particularly, there are increased heterogeneity of some proliferation cells as measured by the Ki67 proteomics data, by the genomic spatial data, suggesting that on a 2-week on treatment sample, we are already able to identify some persistent proliferating endocrine therapy cells, which may be able to help us to actually identify additional molecular targets. In that context, when we look at the spatial proteomics data, by looking at these spatial tumor cells, we identified that there are overexpression of immuno-oncology targets, protein pathways, overexpressed among the poor responders, and as well as PI3 kinase. So this presents quite a nice new data and insight among looking at this very rare kind of big cohort of ER-positive, HER2-positive cancers that hopefully that can help us to actually study a little more in terms of this subset of samples.